Hi, I'm Nathan Hillson, and this is the third part in a series of JBEI software tutorials for its ICE software. In this part, we're going to be looking at some of the additional types of information associated with entries in ICE. For example, who's been looking at this particular entry for DNA sequences? Does it have any sequence validation information for it? Does a particular microbial strain have any experiments associated with it? So that's what we'll be checking out here. For example, one of the things that we might be interested in if we were looking at a particular plasmid is, has its DNA sequence ever been validated? So kind of up here on the top right, in addition to general information, we can kind of click on sequence analysis. And in this particular pane, we can see that we have 4 different Sanger traces that have been uploaded. What we can do is we can visualize each of those 4 Sanger traces as arcs kind of around the perimeter of the plasmid. And where it's green, it's good, and where it's red, there would be a particular mismatch. So if we want to get a little bit more information, you can kind of click on one of those arcs. And if we scroll down, we can basically start going through the actual alignment of the expected and the query, and then you can start to see where there might be insertions, or there might be transmutations, and so on. So you can basically see that for each of the 4 different arcs. So in addition to kind of sequence information that we might be looking at, you might also find some comments that might have been made on this particular plasmid. So there aren't any particular comments for this particular entry, but there might be. And you can also see the history of who's been looking at any particular entry if you're the owner of that entry. Now, another little feature that we want to kind of show out is if there's any experiment data that's been associated with this entry. So I'm just going to search for a particular strain. And in this particular strain, we can see that there's actually some experimental data associated with this strain. So if we click on that experimental data, now we have a list of experiments that have been performed using this particular strain. So these are links to the experiment data depot. So if I open that in a new tab, now I can basically get to the experiment data that was derived using this particular strain. So that's pretty much it for looking at some of these additional pieces of information about the sequence analysis, or comments, history, and experiment data. So that's how you can see if there's additional information associated with different entries in ICE. It's experimental data or sequence validation data, who's been looking at the entries. In the next part of the series, in part 4, we'll be looking at how you create your own entries in ICE.